Thank you again for being here at this amazing conference. We've had a wonderful morning of technical sessions, and those of you who are in the plenary session now have a better idea of global medical technology, a really fascinating topic. Now, many of you were in the 5-minute pitch competition this morning, and those that weren't are probably curious about it. So I'd like to call to the stage Paul Rothweiler and Bill Benton, who ran that session, and they will be telling you who the winners are. So come on up, Bill and Paul.
I left my glasses. Gwen, can you hand me my glasses?
Gotta get the right winner here.
Well, thank you very much. We got a chance to hear from 7 great projects, and they did a great job of presenting what they'd been working on. It was a very tough decision by the judges, almost historic, I was informed. And so it is our privilege to be able to announce the 3 winners in the category. So, in third place in the 5-minute pitch competition, it goes to Thomas Lillyholm, and he was awarded third place at the meeting today. So thanks very much, Thomas. We'll get it figured out. This is the first of many. Yeah. Thank you. Congratulations. Thank you. And I think there's another award that goes with it or something, but we'll fill you in. All right. Very good. Thank you. Second place? Thank you very much. And in second place, we would like to award it to Connor Huxman for his presentation. Connor, please come forward. You're young, you can jump up those steps. There you go. Thank you very much. You can open it up and show them. There you go. There you go. Thank you. Congratulations. Drum roll, please. Yes, we go. And it's our privilege to award the first place grand prize award to Catherine Bradshaw. Catherine? Good job. Thanks very much. That concludes our part of the presentation. Thank you.
Hi, I'm Matt Johnson. I was one of the judges for the Student Design Showcase last night, and you can see we had a number of judges, so thank you, judges, for judging last night. We had 18 student design groups that came in from 11 different colleges and universities. It was a wonderful display of talent and innovation. As Will Durfee said last night, we're in really good hands for our future of med tech. Of those 18, they were all fantastic, but we had to award 3 winners. And in third place was the University of Minnesota's ultrasound transducer positioning aid for fetal heart rate monitoring. I'm not sure if they're here, but if you are, please stand and be recognized. Second place was the Drippy, a gravity bag feeding rate modulator. I think I saw from University of Pennsylvania. I saw that group. And the grand prize award was from the University of Iowa, adaptable below-the-knee pediatric prosthesis. If you're here, recognize it. So, congratulations. It's my privilege to introduce John Bischof, who's the Distinguished McKnight professor of mechanical engineering and the director for the Institute of Engineering and Medicine at the University of Minnesota, who will introduce our keynote speaker. Thanks.
Thank you, Matt. It's a pleasure to introduce my friend and colleague, Dr. Rashid Bashir. He's the Dean of the Grainger College of Engineering at the University of Illinois at Urbana-Champaign. He's a true science renaissance man, and he holds tenure appointments in UI's Department of Bioengineering and affiliate appointments in electrical and computer engineering, mechanical science and engineering, material science and engineering, and molecular and integrative physiology. After earning his Ph.D. in electrical engineering at Purdue in 1992, he worked at National Semiconductor for 6 years, then returned to Purdue for 9 years as a professor of electrical engineering with appointments in biomedical engineering and mechanical engineering. He moved to the University of Illinois at Urbana-Champaign in 2007 and has held a dizzying array of positions since then: professor of electrical and computer engineering, professor of bioengineering, director of the Micro and Nanotechnology Laboratory, co-director of the Center for Nanoscale Science and Technology, head of the Bioengineering Department, executive dean and chief diversity officer of Urbana-Champaign's new Carle, Illinois engineering-based College of Medicine, and since 2018, the 15th dean of the Grainger College of Engineering. He has co-founded 3 startups and published over 250 articles on biomems, lab on a chip, nanobiotechnology, and applications of semiconductor fabrication to biology and other fields of biomedical engineering. He was also recently inducted into the National Academy of Medicine. Let's all please welcome Rashid to the stage.
All right, good afternoon. All right, good afternoon. Okay, thank you. Thank you all for being here. It's really an honor. Thank you, John, for inviting me to give this talk. When John says you should do this, I do it. So I think I'm not holding up to meeting all of the advisory board meetings, but it's really been great to know John and many colleagues here. There's a lot of exchange between University of Minnesota and University of Illinois. Our chancellor used to be here for many, many years, and also your current dean of engineering, science engineering, is from Illinois. So today I'll talk about actually my research, really, and some of the work that I'm doing. I won't talk about the College of Engineering. So we have just a phenomenal team at the college and in my research group that still allows me to be research active. So what you'll see here is really today I'll just talk about kind of what we're doing on the research front. We are a big college. I'm sure you know, so we're now about over 10,000 undergraduates in engineering that are getting engineering degrees, and about 6,000 graduate students, about 480 tenure track faculty, and a lot of exciting things going on, and many collaborations. And we're looking to grow those collaborations across the Midwest with partner institutions. So today, I will really talk about some of sort of this intersection of engineering, and biology, and medicine. So this was a perspective article that I had the honor to participate in with some giants in the field of bioengineering, Professor Shu Chen, who is at UCSD, Bob Nerem, who was at Georgia Tech and has passed away. He was a great mentor for me, and Roderick Pettigrew, who is now the executive dean of the engineering-based medical school at Texas A&M in Houston. So anyway, I think this is, of course, preaching to the choir, but how do we get engineering and medicine to really integrate more and more together? And we posit that this integration of engineering into medicine and medicine into engineering, until these boundaries vanish, will play a very critical role in achieving these broad and specific goals. So again, I think this is what we all believe in, and we're going to keep pushing forward to bring these fields more and more together. Right? I do believe that as we move from quantitative biology to quantitative and precision medicine, to starting to really quantitatively think about health outcomes in the medical practice, the medical education must change. So this is just a short preamble, a short overview of our College of Medicine. So the basic idea that most medical curriculums, pretty much all medical curriculums, are based on biological sciences and clinical sciences as the 2 pillars. This is back from Flexner report from early 1900s. It's an interesting report. I would welcome you to look at that. I'm sure if some of you have seen it. It's been revised over the years, but Abraham Flexner was actually also the person that started the Institute for Advanced Studies at Princeton much later. So he was a visionary, and he was charged by the Carnegie Foundation at the time to review medical curriculums. So long story short, that's that's been the basic, and I mean, our basic thinking was, as I say, that just like, I mean, you know, AI will not replace physicians, but I think physicians who don't know AI in the future might be concerned. maybe it's something we need to teach everyone. So what we did basically was to integrate this component, a third pillar of engineering and innovation, into the medical curriculum. And we didn't have a medical school at UIUC at the time. It was a regional site of the UIC College of Medicine. We wanted to do something different and innovative and what was really needed. So this is where we then came up with the Carle, Illinois College of Medicine. Now we've graduated 2 classes. The third one will graduate this year. Mark Cohen is the second dean. It's received provisional accreditation. And the basic idea is that really engineering and computing is integrated all through the 4 years. So every course has cases, and every case then has a fusion of basically every course director is really 3 people. There's 3 people that are co-directors, and you have a basic scientist, a biologist, chemists from various departments across the campus, a clinician from Carle Hospital, and then a computer scientist or an engineer. And the 3 individuals develop the material together, essentially. So we came up with this new curriculum, which is based, obviously, it's an organ system-based curriculum, but it cuts across and has innovation, computing, and built in. So that's something I just wanted to share. I'm very excited about it still, and as a Dean of Engineering, we're trying to kind of support that. And I say the story that I wanted to go to med school at one point in time and couldn't go for a whole host of reasons, and later I get to help start one. So it just was so special to me personally, and that's, you know, you never know where life will lead you and how things will turn up, as long as you keep trying. So costs of healthcare are unsustainable. This is actually an older slide, but just the bottom line is that how do we work towards this triple aim, right, that all of you know, higher quality healthcare for more people at lower cost, right? So this is called the triple aim of healthcare. You know, can we come up with something like a Moore's law for healthcare? How do you actually reduce the costs over time? And this is a very complicated, of course, question. I think many of you are much better equipped at answering that. I'll try to, you know, propose or present a couple of technologies that I think help us address these challenges, but the problem needs lots and lots of people to work at. And I think now, you know, there's a lot of exciting things happening, AI in medicine, thinking about, of course, getting ready for a future pandemic, understanding infection and being ready for it next time. Health disparities and rural healthcare delivery is a major challenge and opportunity. I still think unlocking the brain and the aging, really aging, and then hence understanding more the biology, the technology, and the psychology of the brain. Actually, around that front, we started a degree program at UIUC. There's 3. There's a psychology degree in brain and cognitive science. There's a neural engineering and a neuroscience, which was there. But the 3 aspects of the brain, so bringing that together in 3 undergraduate degrees that work together. So I think it's a very exciting time for this field, right? Just my last slide on kind of things that we're doing at UIUC that I just want to mention. We have this Carle, Illinois College of Medicine, which is this engineering-based College of Medicine. We also have created a cancer center, which is engineering-based. Technology and engineering is a core piece of that cancer center. And our director there, who's a bioengineering faculty, is now working hard to get NCI Basic Science Cancer Center designation. We have a healthcare engineering systems center, which is a partnership with OSF Hospital in Peoria, Illinois, which is about an hour and a half from us, but a donor actually provided funding to bring engineers and physicians together, and now that's about a $100 million endowment, equivalent endowment, the income of which is used for translational projects. We have a Mayo, Illinois alliance right in your backyard, so I think there's more reasons to want to work together. And then we're trying to, of course, grow our bioengineering department. I think at the end of the day, you have to have a strong bioengineering effort to grow engineering in medicine as well. Okay, so with that, let me jump right into kind of some of the work we're doing in my group. And I'm very, very interested in using nano, micro, and macro technologies, developing small devices and materials and building things, and then applying it to grand challenge problems. So half of my lab kind of works on tools for precision and personalized medicine. I'll really mainly today focus on this issue of sepsis and pathogens in body fluid. And then hopefully this time, and I can show some slides on our 3D biofabrication work, we're also trying to build 3D biological structures that can be potentially used as machines, now thinking about forward design of biological machines. So let's talk about sepsis in critical care in hospitals. Sepsis is a big challenge and a big problem at a very basic level caused by some sort of an infection or an insult to the system. Can result in this dysregulated host immune response to infection. And then there's a whole cascade of events that happen. I won't go into the biology of it. It's a very complicated hyper-inflammatory and an anti-inflammatory response that can result, if not caught early and done something about it, can result in organ dysfunction, failure, and a very high mortality rate. This is numbers as of 21, I think it was, or 21 or 22, you had 11 million people that died of sepsis, COVID was 7 million, and cancer was 9.6. So 1 in 3 people in hospitals in the US actually die of sepsis. They have some other condition as well, the immune system is compromised, and then get an infection. So 1 in 3 people in hospitals die of sepsis. It's a huge problem. Again, as I mentioned, it's actually the leading cause of death in critical care in the US and the world, and you need fast, accurate identification. And what you want to do is try to do, of course, if it's an infection that you can do something about, you want to give antibiotic therapy within just a few hours. That's really, really important. Today's workflow, just very quickly again, there's onset of infection, patient feels abnormal, you might go to the emergency department, takes a little bit of time there, a blood culture is ordered. Blood culture can take up to 5 days for a confirmed negative. Right? As soon as blood culture is positive, you can do PCR or some other type of genetic or nucleic acid identification. And then if the labs have the capability, might do an antibiotic susceptibility, and then of course the patient is in the hospital or not, and there's treatment. But there's some big challenges. First of all, this blood culture, and I'll talk about that in just a little bit. It's so interesting that this is such a basic and cheap technology, and there's been no replacement of it till today. There's been no replacement of blood culture. If you want to grow the pathogen, you have to do a culture, and that can take many, many hours, because the doubling time, for example, for E. coli is about 20 minutes. For Listeria is about 40 minutes. So these pathogens take time to grow, and you do a blood culture. So you collect, and what happens is the mortality risk is increased by about 7.6% every hour during that septic period. So it's something you really want to detect early. Also, blood cultures take a long time, and it's very interesting. This is a sampling problem. 50% of them come out actually negative, even when there is an infection. The person is clearly sick, visibly sick. Sometimes the infection is not caught in the sample that you are taking. The question also is how to quantitatively profile the patient's immune system, and then, of course, how to prioritize and stratify these sepsis patients and the antibiotic treatment, right? And also, this is another huge problem. We all know where physicians are giving too much antibiotics. So this antimicrobial stewardship is really a very important thing, the systematic effort to educate and persuade prescribers of antimicrobials to follow evidence-based prescribing in order to stem this antimicrobial overuse and this antimicrobial resistance. This is also a huge, huge challenge. Today, all of us, those who have gone ourselves to the emergency department or urgent care, you have this, you want the antibiotic. Well, most of the time you don't need it, but we all want it. So the idea that can you take a sample, body fluid, could be any of these sources, rapidly detect the pathogen, and then do the drug treatment. Also, each person's immune system is different. How your immune system will respond to that infection is different than others. So could we also profile the immune system by looking at cell markers or proteins or nucleic acid from the blood? And as you do more and more of this, what's also now in this day of AI and machine learning, you can actually, if you can collect that data and keep it, then you can do a lot with sort of this training data and the feedback for the next iteration and actually be able to stratify the next response. So we are very interested in building devices and looking at sort of micro-nano scale phenomena. So on the left, it just shows, I'll try to point here sometimes and sometimes there. Kind of trying to build various steps, various processes for biomedical lab processes on a chip or a microfluidic device. And the kind of things that are done in the labs are, of course, the pathogen culture, identification, hematology analyzers, blood cell counts, and perhaps in some cases flow cytometers or looking at specific markers, which most of them is really not part of a clinical practice or clinical labs yet. Right? So how can I take these steps and actually translate them to make them point of care, provide faster results and more sensitive results? So let me talk about them. Depending on time, I'll go through some of them. So I think this is interesting. We actually followed these samples in a lab in our partner hospital at Carle, and the lab directors were very helpful in making the students and I truly understand how this is done in the lab once you collect a sample, right? So I'll just go through one of them. So when you are looking for, let's say, if looking for bacteria is a goal, there is 5 mLs of venous draw. That's one of those tubes that are done, and maybe you'll do multiple draws. And then you basically send it for culture, which is run for about 5 days to get a confirmed negative. If it's positive before that, then you stop, and you take a sample of that, and you run it through, you actually run it through a purification and extraction of the RNA, DNA, and then you run it through a PCR or some sort of a nucleic acid amplification. So in this case, this culture is really important because the amplification ensures that there is enough pathogens so that when I take a small volume, about 20 microliters, let's say, per reaction here, I'll guarantee that there is at least a few bacteria in that small volume. So the beginning concentration that we talk about culture can be as sensitive as a few CFU per 5 ml, meaning even a few pathogens in a 5 ml tube, but it's because you're doing that culture, right? And the same thing applies for fungus, yeast, and molds. Virus, there's a separate problem. You can't culture it. So the concentration detection is much higher because you have to have that high. So many times these infections are viral and they cannot be detected unless the number is very high. Okay, so the reason this is also a problem from whole blood is because there is all sorts of inhibitors in the blood that slow down or stop these reactions of the polymerase. So there are non-specific binding of entities, for example, hemoglobin, IgG, that actually bind to your target DNA or even the primers, and they will basically inhibit your amplification. Similarly, same thing, IgG or hemoglobin or heme would affect the activity of the polymerase. So you cannot run PCR directly from whole blood. Extraction and purification is used today, okay? And this is a commercial devices, commercial kits that are available. The idea here is that you have inhibitors that you want to remove, and if you have target DNA, as you do more and more treatments, you also lose some of that target DNA. So the challenge is to do the extraction of inhibitors at the same time not lose the DNA target that you're going after, okay? And the bottom line is that the recovery then is very low for low number. If the initial the DNA is low, then the recovery actually is low and you lose that DNA. So how can you get, bottom line, how do you purify DNA from whole blood without losing efficiency? And can we skip the purification and culture step completely? So we have some ideas on this. Some work has been ongoing where we use this idea of drying the blood. So actually, if you take whole blood and you actually dry it with a thermal step, you end up creating 2 phases, a solid phase and a liquid phase. The solid phase has the nucleic acid of the target pathogen, and I'll show you data in just a little bit. You create this material that has micro nanoscale structure the enzymes can actually diffuse in and get to the target. At the same time, what was really very interesting to find is that the background inhibitors actually get locked in that solid matrix. So you actually inactivate the heme and the IgGs and other inhibitors, whereas then you can do this amplification step. We call it a biphasic nucleic amplification. We have done it with LAMP, which is an isothermal step. And as a result, what happens is that the amplified product goes out into the liquid phase, and you can get a very clear signal. So this is, for example, whole blood, and this is the dried blood, which is then once you add some fluid, you have this biphasic matrix. So this biphasic method, we think, can be very, very interesting and impactful to actually try to help us bypass culture. So there's a phase separation, essentially. Enzymes and primer diffuse into the solid, into the porous matrix. The amplified product diffuse out. Inhibitors are immobilized, and fluorescence reading can be done without interference. So we are pretty excited about that. I'll kind of skip through some of this, and it's pretty detailed. The bottom line here I want to show you is that we did then mass spec on the solid matrix and proved that the inhibitors are actually present in that solid matrix. So they're locked into the matrix. And inhibitors become physically locked, and there's no elution to the supernatant. So this is something very, I think, interesting and exciting. And the goal then is to do this as a way to essentially bypass culture. So then we did a study with clinical samples. Here, this is E. coli pathogen at 1 CFU per 800 microliters. So in this case, we haven't scaled it up to 5 ml yet. This is 800 microliters, which is close to the volume that's taken from infants or little kids. You won't take 5 mLs from little kids. You take about an ml. So this is about 800 microliters. And here you can see we can detect as few as 1 CFU per 800 microliters, 10 CFU, 100 CFU. This is the threshold time for detection. And this was then a clinical study in the sense that we would get a sample of the blood that the lab would order a culture. And when we found that the lab found a positive, we would check it for that pathogen in our blood sample with our technique. And this is what we found, essentially, that we can have these E. coli. We detected E. coli, MSSA, and in this case, some samples were negative with MRSA. So we were able to get results in an hour and a half, less than 2 hours, whereas the culture obviously took, in this case, in many cases, multiple days. So this is, I think, pretty exciting, and we're going to kind of continue to push this forward as a technology. Now, how do you scale this up? So this is work ongoing. We want to, think of taking the large volume and splitting it into a large number of small reactions, whether it's a 96 well plate or a 384 well plate, and to think of that further, you would really want a sort of microfabricated or an additively printed cartridge. Think of it as a pixelated petri dish where blood can get filled in and essentially go into these small chambers where each one of them has this drying reaction happening, and then reagents are introduced. So this is work ongoing, but we're very excited about this possibility of trying to move this technology forward. Okay. But if anybody, I think, clearly has any ideas on or needs to work, it's elimination of blood culture. If you can figure out a way to bypass blood culture and directly extract the nucleic acid and be able to detect a panel, like any one of X number of pathogens in a panel from that starting small amount of DNA, this would be transformative. This is still the gold standard in the lab, and it's just taken for granted. Okay. Let me skip to the next couple of things, a couple of other technologies. So now point of care, sort of nucleic acid amplification. So we all obviously learn how to do a swab and learn how to even give saliva samples, spit. So this is that March 2020 when the genome for SARS-CoV-2 was released. We actually very quickly developed the primers for LAMP amplification. They weren't available at the time, but looking at the genome sequence, you can actually design the primers. And we were able to collaborate with Professor Bill King and Brian Cunningham, that many of you know, and develop these additively printed cartridges with a cell phone reader. And I think all of these technologies moved really quickly. They got a lot of push, right, during COVID. But we were able to demonstrate that we can do this from a swab, from a clinical study using patient swab, and using a cell phone camera, you can get amplification of LAMP. And I think these technologies have already been deployed by a few companies, and there's no reason why these technologies cannot be commercialized at this point, even more so, and actually lower the cost even more. We have developed multiple generations of this. This was the very first one, but then the second one, we actually did 2 strains, the N and the S gene, remember at the time when we were looking for different genes. And then the most recent one, we can actually do 4 targets plus 2 control. So this paper we are just about to submit, but we can do flu A, B, RSV, and COVID all in one cartridge. Now, what's next in this domain, right? So I think LAMP or PCR, this we can do now. There's commercially available point-of-care examples of this now. What's next is really if you can develop a molecular amplification-based test that can provide results in antigen testing times, so in less than 10 minutes, at the cost of an antigen test, right? So lower cost and results in less than 10 minutes. So this is something that we actually developed a technology, and it's being commercialized through a company called Veda Bio. I can look this up in the news. So this, and I wanted to bring this up because here we did things very differently. We actually didn't publish the work, but rather patented it first, very actively, some through the university and some through the company. And then the publications will come out now. So this was an interesting example of kind of this innovation and translation, where in the university we're incentivized to, publish, but at the same time, I think many of you know and have done this, there is a way to do this in a balanced way where you protect the IP first and then publish after that, as long as the funding agencies and as long as the students are not getting compromised right through that and getting their educational experiences. But in this case, so the idea was it's a CRISPR-based method, and you can look up the patents, which provides a 2 system. There's 2 CRISPR systems, and it's a cascade reaction. It's an amplification reaction without enzymatic activity for amplification, but rather 2 CRISPR systems that actually give you a signal from a very low number of starting molecules within about 5 to 10 minutes. The time here is important because if you are in an urgent care, you don't want to wait more than about 10 to 15 minutes. So if you can truly identify whether my daughter or son had a viral or a bacterial infection from a sample within that short time with a high sensitivity, I think that again would be very transformative. Okay. The last thing I want to just briefly mention, this technology that we've been working on for many years is this idea of measuring, again, characterizing cells and protein biomarkers or even nucleic acid biomarkers from a blood sample. So we have come up with this approach some years ago, which is actually a differential Coulter counter. I think everyone has heard of Coulter counter, which is an electrically based method to characterize and count cell. So we make a microfluidic device which takes whole blood, lyses the red blood cells, recover the white blood cells, count them at this first counter, then have a capture region with the highest surface area to volume ratio. And this technology, this module is similar to the CTC capture from Toner's group et al. Here we capture the cells, and then we count again what's going out. So then the differential count here can also tell you what's been captured, and it's all sort of at a point of care and electrically based without the need for optics and lenses or cameras. So using this over the years, we have developed and we have shown you can do total white blood cell count, but also lymphocytes, monocytes, granulocytes, CD4, CD8, CD64. And what's very exciting is that using the exact same platform from the same sample, there's a potential for also doing proteins and DNA markers. So the devices look something like this. You have whole blood coming in here, and this is sort of the classic textbook microfluidics where you have sheet flow coming in. This center stream is the blood cells, which get lysed after a few turns by diffusion of the lysing solution into the central stream, and you can see that basically the solution becomes transparent because all the red blood cells are lysed, and the white blood cells are recovered. You have to stop the lysing by adding a quenching buffer. So using this, then, we also demonstrated that we can capture CD64 expressing neutrophils. So CD64 expression on neutrophils is a marker for infection and for sepsis, actually. The higher the expression, it's been correlated in literature that patients who are in more advanced stages of sepsis have high expression of the CD64 on their neutrophils. So here, at the bottom, these are actually clinical samples that the x-axis is the control measurements from a flow cytometer, and then the y-axis is a sample of blood from the same sample that we take, and then we use our devices to measure that. And you can see a reasonable correlation here, again, from devices that were built in our labs, so, and all run by students. But here we had to optimize the shear stress at these chambers wall. That's how these microfluidic devices for this cell capture, you have to optimize the flow rate and the geometry to be able to capture the target cells. And we also, as part of that study, showed that using the devices, you can actually track the CD64 expression as a function of time for that one patient. So over time, you can track this as well. So this again could be useful as a marker for sepsis, right? And then using this same technology, we can also do proteins and DNA. Now, how would you do that? Because cells are much larger, and in the earlier study, we were counting cells. So the way to do it is to actually use beads that are about the same size as the cells. Using the beads, you would capture the proteins or DNA, and now you count the beads that are going in, and then in the capture chamber, you have the complementary molecule to be able to capture the beads when it has a target. And then you count, again, what's going out. So using a similar differential approach now using beads, you can also measure cells or proteins. What is interesting here, we needed a new technology to be able to multiplex this. You needed a different electrical signal from each bead. So the way we did that, we actually collaborated with Professor Heeson-Han in chemistry, who can synthesize these beads with different gel composition and different, hence resulting in different electrical properties. So essentially, you have beads that are the same size, about the same size, but give a different electrical signature. And that way, we can actually multiplex this and detect different proteins or different microRNA, for example. So we demonstrated this for proteins and for microRNA, that you can actually detect high and low concentrational differences using that same basic core technology. So that's very exciting, because I think if you can take there's nothing today where you can take a blood sample and count cells and measure proteins, for example, from the exact same sample. Okay, now let me bring it all together. I know there was a lot of details and a lot of different technology modules. So where we're going with this, the goal was, sepsis. What we did then, we pulled a big team together from across Carle, and the idea was to do a clinical study. So as we were building these devices, we were very excited about the devices. But then what Bobby Reddy, who I'll highlight again later, realized is that as we were getting the samples to test our devices, we said, well, getting these blood samples, this is really powerful. The real value here is in the samples, because there's so much more in there, and these samples are being discarded. So in hospitals all over the world, everywhere, when the blood draw is done, you use a little bit for the testing, and then the rest is thrown away. So we got a study approved, IRB approved, to get the discarded sample. And as we started to collect that, we realized, the power of whatever we're trying to do is not in the devices, it's actually in the sample. There's so much more information. So we looked in the literature, and there are known proteins that are pro-inflammatory and anti-inflammatory. So we said, okay, we want to measure all of this using today's technology, which is ELISA. And we would also the hospital agreed to give us the entire data set for that patient. So we started to get the EMR and the blood samples for each patient that a blood sample, that a blood culture was ordered. So here's data, and in the last few years, everything that's happening with AI and machine learning. So we collaborated with a couple of statistics and faculty who were working on AI and machine learning. We learned ourselves this. This was all new for many of us who were working on devices all these years. So long story short, we actually used 5 machine learning algorithms with and without various feature selection and mechanisms, and estimated their prediction using these repeated tenfold cross-validation. You had to get this education from the physicians and the residents, because there's no gold standard. So they had to look at the charts and sort of figure out what state the patient is in, and they classified them into these 11 stages that are at different time points of the patient's progression through the hospital. And then long story short, this is what you come up with. You come up with these normalized feature coefficient, which basically points to the importance of that parameter to the end outcome, which is whether the patient will be discharged or the patient is going to progress through the septic stages. So these are all the proteins that were measured, and then there's also some other EMR parameters here that are taken. But you can see that there is no one biomarker for sepsis. It's actually multiple ones, so IL-6, PCT, MMP9, there's multiple parameters that are actually highly correlated to the outcome. So this is kind of the bottom line here. So then if you look at the predictive power of these parameters to the end outcome, what is really, really interesting is that these two curves, the purple and the orange, is today's current clinical practice. They use these parameters called SOFA and QSOFA, which is a very simple algorithms that if the physicians have time in the ER, they would use this algorithm to come up with a score and then stratify the patient. So you see that the predictive power is actually 0.5. So what does that mean when it's 0.5? It means it's a coin toss. It's 50% chance that you are right, 50% chance you are wrong. So that's what's happening today in the clinics. If you start to add these parameters, you can see that the predictive power starts to go up, and you can approach 0.8. And now this data is, again, a couple of years old from our lab, from research, working with the company and with Carle, and now this has been advanced much more. But this actually then resulted in a company called Prenosis. And the CEO is, and co-founder is Bobby Reddy, who was actually in my research group, but then has really worked. I mean, this has been his life, and I give him all the credit for taking the company where it is. And it's really exciting, as a matter of fact. So the idea is that you'd have a big data set, you have a device, a person comes in the emergency department, you measure a few parameters, maybe 5 or 6 proteins using this point-of-care device, and the data that's measured, it's quickly compared to this very large data set that's there. And then there's a decision support system, essentially, that informs the physician at the time of a priority score or how much to pay attention to which patient. So this actually, they have now the world's largest biobank that's linked to clinical data for infection and acute care. They call it the Immunix Precision Medicine Platform. And as a matter of fact, last week, I don't know if any of you had heard this, but we were super excited. Last week, this was the first AI algorithm that the FDA approved for sepsis stratification. So this was really great news for them. He's very happy. He's trying to figure out, of course, a lot more work to be done. But this is an example, again, of these technologies coming together, but also things going in different direction. We started out with a device, thinking that's going to be the core of it, but really it ended up being the data and the sample and the power of what's in that sample. Okay, so one other thing I'll mention is, we're all trying to also detect molecules at a much higher sensitivity level. So just very quickly, a project that we worked on with collaborators in mechanical science and engineering and material science was use this graphene. I think many of you might have heard of graphene as a 2D material, and it can be used as a field effect transistor, where you can detect the charge on a DNA molecule when it's close to the surface of a transistor, Well, it turns out that if you could crumple that graphene, if you can curve it, essentially, shown here, and this is attached to a polymer substrate and then shrunk, and as a result, you get this very crumpled graphene. What happens is that the debye layer can be modulated, and you can actually extend the debye layer in these concave regions so that the molecules there can be detected with much higher sensitivity, meaning much lower number. The sensitivity becomes much higher, and we were able to show that you can actually detect a few molecules directly in about a 10 to 20 microliter drop of volume. So very sensitive detection using this crumpled graphene. And this, I mean, the hope here is that something like that could directly detect the molecule and you don't need the amplification at all. So those are the kind of things that we've been working on. I want to maybe show you one other area. I think I might have maybe 15 minutes or so. So thinking about tissue engineering or sort of building things, with living material, there is developmental biology. There is, of course, tissue engineering, which is reverse engineering, biomimicking, trying to replace, repair organs. But what if we were to start thinking about forward engineering and building systems and machines and devices that are all made with cells and soft materials? So this idea of sort of engineered living systems using 3D printing, 3D bioprinting. So these printers are now everywhere. I saw many today in the tours. We took a commercially available system and modified it to print hydrogels with cells embedded. Also developed a micro-scale stereolithography apparatus where you can turn a CAD drawing into a part. So this is now commonplace technology, right? But adapting it to biological applications to actually create hydrogels that are wet, or maybe can grow cells on it or cells in it, I think it's something also very exciting, and there's a lot of work happening in this domain. So we have been kind of working on that for many years. But where we went with this is to try to create sort of modules that can be used as building blocks for things yet to be imagined. So we were able to create these muscle strips and muscle rings using a mouse skeletal muscle cell line. We can 3D print this with fibrin, collagen, matrigel, and actually create these different shapes. These are millimeter to centimeter scale tissue. Here, what you see here is these muscle rings that have been formed, and then they can be taken from one scaffold, from one mold, and moved to another one. So they're pretty robust, and now multiple labs have repeated this. So this is a muscle ring. Let me go back, sorry. And we can also do the same thing with the neural tissue that I'll show you again in a little bit. But the bottom line is using this, you can create these miniature robots, or we call them these biobots. So what you see here is a side view of one of these structures that has two pillars, call them legs. The whitish region here is this muscle ring or muscle strip. And in this case, these cells were optogenetically transfected with channel rhodopsin. So they actuate when you shine light. So what you see here is every time you shine light, they're sensitive to blue light. You shine light, the muscle actuates, and the structure is designed to break symmetry such that it actually moves and walks in one direction. So it started out here, and every time you shine light, it actually inches in one direction. So this is walking in fluid on a surface of a petri dish at this point. So it becomes very interesting. We actually turned this into a lab, into a junior level lab in bioengineering, where students do the cell culture, 3D print, and they can make whatever different structures they want, we know the material properties, so we know the K, we know the X, we can measure forces, we can do some very basic characterization, and essentially, you can get you can see displacements here that are on the order of a body length in about 30 seconds. So you can, see this move. So again, it's a biological actuator that you can design you can forward design, right? You can then make structures that are moving in different directions. Here's one that is going from right to left. It's got 2 muscle rings. So you shine light here, it's going to move in this direction. You shine light here, it's going to move in that direction. You can also make some that are 3, can go like, this has three muscle rings here. And if you shine light on this one, it'll move down in this direction. Sorry, I'll move this, yeah. Okay, anyway, you get the point. That it's going to move in the direction you're shining the light. So you can get this optical control of these biological structures. And then actually, most recently then, we actually collaborated with John Rogers' group at Northwestern. So he's got this really cool flexible electronics. So his students designed this micro LEDs that are wirelessly controlled, and we designed our structure to integrate this into this. So here now you have 2 sides. There's a central region where this circuit sits in, and it's got these LEDs hanging out on the side. So you're actually stimulating now with a local LED, and then using that, you can actually get this thing to walk and even take turns, because it can shine. The frequency on one side can be higher or lower, and it will actuate more, and it can turn in a maze. So it's kind of walking in a maze. And here's another example of that. So this is actually in fluid with muscle cells that are being actuated with light, with the micro LEDs, remotely controlled. So again, this whole space of sort of biological actuators that are embedded with electronics, and maybe we can use it for non-engineering applications, right? So there can be some very interesting possibilities in this direction. This was the video I was going to show earlier, but that same muscle compaction process, you can also do it with other cell types. So this is with neurons, and we have a training grant, an NSF training grant on sort of miniature brain machinery, we called it. So here we can create different shapes. This is a ring, this is a rod. These are, again, optogenetically transfected mouse embryonic stem cell-derived motor neurons. And here you see, look at this bottom one, you see a ring. This is the tissue ring. We're shining light on one end, and we are measuring the electrical activity on the other end using an MEA. And when you shine light on this side, you can see the activity on this side increases, essentially. So pre-stimulation, this is the firing rate. Post-stimulation, the firing rate goes up downstream. So those cells are connected, and you can now start to think about programming it, start to interrogating it using electrical and optical signals. And the goal where we're going with this is unpublished work. We want to make a neuromuscular junction. So those biobots should then be, now muscle should be driven by a neuron. And this is actually very recent results that are not published, but this is there's a neuronal tissue that is driving the muscle, and it's spontaneously firing and driving the muscle. So it's actually moving in one direction, but autonomously in this case, driven by the neuron. So there's some very interesting possibility here from an engineering perspective as well, because can you get these things to maybe learn and kind of know where it's going, or be able to sense and go up a gradient or down a gradient. So I think there's some interesting possibilities there. Okay, the last thing I'll show is just recent. About a year ago, we were also worked with the team with University of Chicago and Northwestern, and we were able to land the Chan Zuckerberg BioHub. This is now led by Professor Shana Kelly from Northwestern. She's the director of the hub. Gene Robinson from UIUC, Jeff Hubble, and myself are on the executive committee. Amanda is our COO. And this hub is focusing on developing technology to measure inflammation biology. So it's around inflammation, infection, immunity. There's different tissue models, and the goal is to develop technologies to measure tissue scale biology in live tissue to understand inflammation. The hub is just kind of, like I said, getting set up. The space is now open. If you ever come to Chicago, please contact them. They would love to give you a tour. The first one was in San Francisco with Stanford, Berkeley, and UCSF. This is the second one, and a third one has been announced also in New York. Okay, so I'll finish by, I'm sure maybe some of you have seen this slide, but I still like this very much in terms of these quadrants of consideration of use and quest for understanding, and this is perhaps more for students, but just thinking about where we land, where is our interest is, where do we want to do work. I think clearly pure basic research is very important, right? We tend to do less applied research in universities, and perhaps we want to do more use-inspired basic research, which is what's called the Pasteur quadrant. But I mean, Edison, right, didn't necessarily care about how why it worked. Tried a whole bunch of things, and things worked. So this has been shown in many different places. But I think it's very interesting in a university. I do believe we need to have the right balance and portfolio of projects that are in this domain as well, and perhaps use-inspired as well. We definitely don't want to lose touch of the basic foundational work just for the sake of understanding, because many great things come out of that. But at the same time, I think having a balanced portfolio is important. And what is that portfolio? What is that percentage? That's for each institution to decide. But a lot of the work we do with clinical partners is clearly very use-inspired. And I think it definitely fits into this as a goal for translation, which is really kind of the, I think, goal for many of us, right? You don't want to be there in the low and low, right? And then finally, this is also another I love this book. I've referred this to John also. This is a really good book. It's called The Usefulness of Useless Knowledge. It's like $10 on Amazon. I would encourage. And it's the reason I ran into this book also because the first chapter is written by Abraham Flexner, who was the same person that did the medical curriculum review in 1910. So when he started the Institute for Advanced Studies at Princeton and brought Einstein and all these people, it was Abraham Flexner that started that institute. But this book has 2 chapters. The first chapter makes his case, actually. He makes a very strong case of the basic curiosity, that the most useless things sometimes are the most useful. So that throughout the whole history of science, most of the really great discoveries, which had ultimately proved to be beneficial to mankind, have been made by men and women who were driven not by the desire to be useful, but merely the desire to satisfy their curiosity. So I think that's a position, very important. The second chapter is written by the current director, and he does make a case still that the sensible approach is to create a portfolio with projects of immediate concern that have concrete applications, and also long-term projects that require deeper thinking. So again, this idea of basic research versus translational research, I don't think it's not a conflict. I think it's just something important that we should be aware of. And nowadays, with a lot of emphasis on solving grand challenges, whether it's the climate, whether it's healthcare, whether it's agriculture, whether it's sustainability, you're also seeing a lot of large-scale funding that are very translational. All of the hubs that you also are working on, the innovation engines, those are all very translational, right? They're not basic science. But universities we are being demanded to do that. And I think it's okay, it's important, because we do need to sort of make that impact on society. But then keep the portfolio, right? Don't forget the basic science, it's really important. Okay, with that, I'll stop. Thank you for your attention. I know I talked for a while and talked lots of details too, but hopefully that was useful. I have a great set of collaborators and students and a whole bunch of collaborators, a bunch of funding agencies. We collaborate with too many people, which is great. Too many people to mention, but a lot of great collaborators, and thank you.
We have time for a couple of questions. If we have some questions from the audience, are there some mics that could go around? So maybe I'll start off. So when did you know that you wanted to be an interdisciplinary, transdisciplinary scientist? When did you know that you wanted to be an interdisciplinary, transdisciplinary engineer scientist?
That's a great question. So I actually went through my Ph.D. in electrical engineering, making semiconductor devices all the way through, like a complete, just in the lab, doing nothing else in life. And I took 2 bioengineering electives in the last semester of my Ph.D. That's when I really kind of discovered my love for biology, and that's what I should have done. So then I went back and actually took 2 years of courses in a community college, organic chemistry, biology, the labs. So then I just was really interested in bringing engineering and biology together at that time. But it was because I hadn't really discovered the biology piece early on, discovered it much later.
Other questions?
You mentioned that universities are getting this kind of pressure because they're you parsing that out?
Yeah, I can repeat the question. The question is that I said that we're getting sort of, there's a lot of interest and, I mean, maybe pressure at times, but expectation that we would also, universities would also work on translational projects. So how do you decide, balance, choose which projects to work on, which not? So I think it's a great question. I think, you know, everywhere the research office is being, like, innovation is being added. It's research and innovation, right? I think entrepreneurship to me is something different, by the way. That's something we need to teach our students. We need to do that. But these larger scale translational projects is something that you have to decide to go after or not, and whether you have the capacity for it or not, whether you can get enough faculty interest, or you want to build a cadre of non-tenure faculty. We have those also. Many of us have research institutes which are all staff now in our universities. So I think, I mean, I personally think it is important. We talk about this a lot in our college. You know, the fact is that the NIH granting agencies, the metrics are still set up for doing the basic work. But I do think that there should be enough of a portfolio to do these major sort of grand challenge-driven projects that universities should work together. They're not going to do this on their own. So work together with local companies, the innovation ecosystem to try to grow that and have that economic impact. So I mean, certainly as a land-grant university, I can say we do feel it's important to have that economic impact. So I think, you know, what is that portfolio, percentages, how, that's I think for us to decide. I can tell you that in our college, out of 300 million annual research expenditures, maybe about 20% is from industry funded, which tends to be a little bit more applied anyway. So I think we're going to keep some, but we're still majority driven NSF, NIH, DOE funding. So, but a great question, yeah.
I'm going to follow up with another quick question here, especially, you know, looking at this array of things that you've done, which is so impressive. It might be hard for someone younger in their career to sort of chart a path through this. And I'm wondering if you can give some advice, you know, because you touched on basic science, the Pasteur's quadrant, all of these different things which you've been able to touch through your career. What would be your advice to, you know, young people starting their careers in bioengineering today? And what, you know, how do they navigate this?
Yeah, this is really important, and I think actually I'm so excited when I talk to students, undergrads and grads as well. There's such an opportunity, right, to make an impact. So I think you're living in a great time when these technologies are exponentially increasing in impact and scale, like AI or these devices. And so I mean, I think what's really important is to have some foundational depth. You have to have some foundational depth in an area that you pick. You have to pick something. I would say don't worry about what that is. Pick something that you do gain deep financial, deep knowledge, right? The financial part will come later, right? I think a lot of times that drives our thinking, but the deep knowledge in a domain is really important. At the undergrad, you can barely do it, but you should try. And don't worry about, I mean, in 10 years out, you will be doing something completely different anyway. So I think the most important thing is to build some foundational depth and learn how to learn. So build a foundational depth and learn how to learn. And then, you know, the rest is hard work, a little bit of luck, and, you know, making an impact. There's a saying I really like, I can't remember who said that, I think it might have been Joe Montana, that the harder I work, the luckier I seem to get. So I think hard work is really important, of course. So coming back, I think a foundational depth in certain area, but then also just this learning process is really important because we're all doing things that didn't exist when we went to school. And now things are very different. So I think the future is bright and we're counting on you all to solve all our problems.
And one question. You talked a little bit about the shift of like first to patent versus first to publish, and I'm curious about are the faculty incentivized to do one or the other, or have a balance approach? Because that is a little bit different at the universities, or depending on the university culture. Can you speak to that at Illinois?
No, yeah, so I think that's a great question, and I think we're still not incentivizing people to not publish, let's say, right? So I think it really depends what kind of the goal is, right? I mean, like in this case, we did what we did because no students were being impacted. So one of my basic principles is that, look, students' progress, whether it's a master's or a PhD in this case, should not be impacted. If they had to make that choice, I would not have done what we did. But in this case, it was postdoc research scientists, it was more in that domain. We said, okay, let's do it this way this time, right? But many times I've filed patent first, at least filed it, and then published it, and many, many times. So I think that can be done if you incentivize that, celebrate it, recognize it. I think we have more work to do on that front. Because we're also kind of very basic science driven. I know our department names are mechanical science and engineering and materials science and engineering. So we're very science driven, which is great. But I think that translational piece is important, and I think we're trying to do more to incentivize, so we just recently created a new translational award in the college. We're doing more things that are recognizing the translational activity and that piece. It's important. But what stage of your career you do it, it's something that we should be discussing and actively managing.
I know I can't project, so I waited for the mic. So you've got so many fantastic projects and companies under your belt with such great teams behind it. What is your suggestions to starting PIs or starting co-founders as they build their teams?
So I think, I have to say when we started this, there wasn't a formula. We just I've had 2 other companies that didn't work. They just didn't move forward for various reasons. These 2, we hope, let's see where they end up in terms of some exits or other things. But I think the most important thing, certainly, I would say even more than the idea, is the people. I think the people, obviously you have to have something that you're trying to, the core idea is there if it's a new startup. But I think the people that you bring together is just so critical. At the end of the day, any organization, I think it's people. If you bring the right people, they'll generate more ideas and stay at the cutting edge or revise the idea that you started with. So in one case, we started with something completely different as a goal and ended up somewhere completely different. So I think people, finding the right team, forming the right team, is just so important. Probably the most important thing in my mind. It's people.
Great. Well, let's thank our speaker once again.
All right. Thank you.
Thank you, Rashid.
Thank you. I'll take this out.